Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes only on YouTube. Little bit my voice would be different because I am suffering from cough. But let's just come back to the major thing, that is to completion of our chapter. So this is a video for 12th class. From the thumbnail itself you must have seen that it is the second part of our chapter Biotechnology, where we're gonna be studying about the processes. In last video we talked about the tools and little bit of understanding about it. Here we are going in depth, full-fledged in a war mode to complete it. So first of all, when we are studying about this chapter, the first and foremost important thing is that you should know that how many processes are there. So there are total 8 processes which we have sequenced up. In NCRT, it might be 6 processes, but 8 processes we have done it so that we can remember the steps clearly. So if you remember these 8 steps, you would be able to remember the whole chapter as it. Plus in NCRT, it has been little jumbled up. So this is what a more synchronized way of studying it. So let's just start it. The process of the recombinant DNA technology. First, we will be doing what? Isolation of DNA. We will take out the DNA. Once we get the DNA, we will do the fragmentation of the DNA with the restriction endonuclease, because we need to cut them up, because the gene of interest might be present in between somewhere, which we need to cut it up, and simultaneously we have to cut the plasmid DNA also. So the second step is that. Now the third step is amplification of gene of interest by PCR, which means what? With the help of PCR, we will increase the number of DNA content. Why we will increase the number of DNA content? Just imagine that if I am working with the DNA, I have only one DNA got with me, which is a gene of interest. Now while working, my colleague just pushed me and all that DNA sample went on the floor. Now what happened? It is lost, or it got teared up, or something happens with that. How I will do that? Shall I repeat all the first 2 steps? It will be time consuming and the cost consuming. Okay, expensive process. So what I will do, instead of that, I will increase the number of DNA fragments into multiple copies so that I can use as much as time I can. Then comes the ligation of the DNA fragment with the vector, because we have to join them. After joining them, we have to transfer it into the host cell. That is called as gene transfer. Then after putting it into the host cell, we have to see that whether the host cell has taken it or not taken it, or what kind of plasmid it has taken up. It is an important one. So this is also we will see. After that, we will see obtain the product from it. If it got the a recombinant DNA, we need the product, means the luminance of light, if you remember the example, or any other product, medicine, whatever we want to make it from it. And when we get the product, we will do the downstream processing, which means we make it for a large scale production. And after that, what we will do, we will check for its clinical trials, its safety, preservative precautions, whatever are needed before to give it to a human consumption. So I hope these are the 8 steps you would have understood. If you would have understood, we can directly hop to the major topic. So one by one, we're going to be studying each step of this process. The first step is what? Isolation of DNA. So there are 2 different DNA. One is the vector DNA, means your plasmid that we need to get it, and second is the gene of interest containing DNA. Okay, and from where we will get it? From the bacteria, or from wherever the cell of the organism from which we want the genes from. Okay, so in this step, what will happen? The DNA which are to be used as passenger DNA and vehicle DNA are extracted out from their cells by lysing enzymes. Remember, we have said about lysing enzymes. So those enzymes will be work over there. Lysing enzymes with lysing the cell with a suitable enzyme. That will be lysing the cell with suitable enzymes. So now imagine that if there is a bacterial cell, we will put what? We have to remove the peptidoglycan cell layer, okay, outside. So we will put what? We will put lysosome, okay. So here I am making what? This is your bacterial cell, okay. So in bacterial cell, we put what? We put the lysosomes. So what will happen? Its outer cell wall will be destroyed. Now, if it is a plant cell, if it is a plant cells, what we're gonna be putting? We're gonna be putting the cellulase enzyme, which will do what? It will break the plant cell. And if it is an animal cell, okay, so animal cells, oh, sorry, not the animal cell, but the chitin cells. Okay, let's take it that fungal cell. So its outer wall will be broken up by what? It will be broken by the chitinase enzyme. So this we know that what we're gonna be using it up. Now the DNA is then, like, if you break the things, still there are what? Different macromolecules are present. Okay. So these enzymes are doing what? These are only breaking the outer cell wall. These will only breaks the outer cell wall, but DNA is intermingled with or enclosed within membrane and have lipids, because membrane will have a lipid layer, okay, have lipid, have proteins, RNA, etc. biomolecules as well. So now we have to remove these molecules also. And how we will remove it? If you remember about an experiment from the biochemical characterization by Avery, McCarthy and McLeod, then you remember that they keep on putting different substances to remove it. Okay, same thing we will also do. Okay, so what we're going to be doing it? Lipids will be digested with the help of lipase enzyme, protein with the help of protease enzymes, RNA by the RNase enzymes. Direct names are there. Nothing so much tough to remember about it. Okay, so what we're going to be doing with these enzymes, these biomolecules will be eliminated and DNA will be extracted. Okay, moving on to the next thing. This is the DNA that has been extracted. It has what? If we put this chemicals, means all of these RNAs, protease and all those things, it will appear as a suspension, like mixed form. We have to separate it out. So we will add what? Chilled ethanol, so that both the things can be separated out from each other. Okay. So now what will happen? We extract purified DNA by treating it with chilled ethanol, and it appears like a fine thread in the suspension. Okay, so this diagram is also being given in your NCRT, that if this is the test tube where you are adding this thing, so what will happen? Let's see. this is your whole thing. This will appear something like this, like this. Can you see this? So it will appears like what? Appears like thread in suspension. Suspension is a type of solution condition. Okay, so it will appears like that, whereas the other lighter this lighter shade, which will be almost transparent, that is what all your dissolved things, these things is dissolved by this and dot. Okay, now the next thing would be that now DNA is then taken out from it. How it will be taken out from it? With the help of spooling process. So spooling is what? It is a long spoon kind of a thing. Okay, and we take it like this and we will spool it up out of it. Similar like what we would be like spooling certain noodles or faluda from the container. Okay, so now this DNA is removed out by spooling method. So this device like or the instrument is like this. This is the spooling spoon, okay, through which we take it out, and its diagram is also being shown in your NCRT for this. So this is about the first step. Moving on to the next step. So what is the second step? Second step is fragmentation. So what we will do, we will divide this whole thing, okay, or the DNA into the parts. So vehicle DNA also will be cut, as well as the this fragment also has to be like DNA, whole DNA where the gene of interest is, when that fragment has to be extracted out. So next step is fragmentation of DNA by restriction endonuclease. By restriction endonuclease. Now what we're going to be doing, both the vehicle DNA and the DNA obtained from the one of the where the gene of interest is present, they will be cut with the restriction endonuclease, and they will be cut with a similar restriction endonuclease, and then we will put them on the agarose gel. Why we gonna putting the all the DNA into agarose gels and mostly the gene of interest containing ones? Because we need to see that whether how many fragments has been made, and we already know that which fragment will have our gene of interest present. Let's try to understand it. So first of all, with the help of restriction endonuclease, purified DNA molecule are cut under optimal conditions. So under optimal conditions it will be cut. Now both will be like, then we will do what? We will be putting this main DNA fragments on the agarose gel. Why are we going to be putting on the agarose gel? Because on agarose gel, when we put the electricity, because of their negative charge present on DNA, this thing we have already know from the molecular basis of inheritance, because of phosphate there is a negative charge present on the DNA fragment. It will act like what? The molecules, negative molecules, which moves towards what? One side to the other side. You know that current if move from this side, the electron moves the opposite side. So same thing gonna be happening over here. So there are gonna be certain things which we will learn it, which we will understand the concept little bit from the electrostats of physics as well. So now what will happen? Then agarose gel electrophoresis is employed is employed to check the progression of restriction enzyme digestion. This thing we will remember, because over there only what will happen if we will see about the agarose gel. Let's take a rough diagram of it. So how the agarose gel will look like? It's like a container box, okay? So okay, so here we have what? Here we have certain wells present. Wells is what? A space in which we will put our fragments of DNA. Okay, so these are what? These are the wells. Okay, it is what? It is filled with agarose gel material. Okay, now we're gonna be putting what? We're gonna be adding the this is what I am making. This is I am making the current flow thing. Okay, so what will happen if I will put the current? Okay, so current is coming on this side. Current coming on this side, and I have put my DNA material. It is here, here, here. Now these are what? This is the DNA which I have put it. Now since DNA is negative in charge, plus there is what? Endo restriction endonuclease has been added to it with restriction endonuclease. So it might have cut different endonucleases we have put. They have cut the DNA fragments into different parts. Now under the influence of current, it will move. Okay, so what will happen? It will move like this. So we're gonna be seeing like this. Okay, same here also. Fragments are here also. Okay, according to their breadth. So here there will be what? At closer towards the well, there will be what? Bigger fragments in proximity. Proximity means closer. Whereas this is what? Small or light. This bigger or heavy. Heavy means it containing more number of nucleotides. Okay, light fragments, they will be what? Distally or far. Why it is happening, we will learn it. Okay, let's just see that something this kind of situation will come. Now the most important thing, we are using restriction endonuclease. Whatever the restriction endonuclease we are doing to make a cut on the plasmid, the same has to be on this. Means your restriction enzyme, sorry, on the DNA fragment also, because the fragments will have a complementary pair which will go and attach to it. Let's try to understand, and from it the major concept also coming up. So concept is what? Concept is both passenger and vehicle DNA are cleaved by using the same restriction endonuclease, so that they have complementary pair, complementary sticky ends. Let's take example. For example, Eco R1. If you remember about Eco R1, what you know that where it used to cut. Okay, Eco R1 cuts at GAATTC. So here and here it cuts. We remember this fact. Let's try to understand this with respect to our process. Okay, so if you see, this is your, first I'm going to be making a DNA fragment. Okay, this is the fragment, and this is, let's make it straight, not complicated. One fragment, another DNA fragment, and simultaneously I'm making what? I am making a DNA like plasmid also. Okay, so 1, 2. Okay, let's just see that over here. If you see, for example, if it is like that, now this is GAATTTC. Here C. I am just writing it so that it's not tough. Okay, TTAAG. Okay, and opposite would be present over here. So CTTAAG, and its opposite is GGG only. Okay. This is here. Simultaneously on this also, there is what? Genes present now. These are making bonds only. So they are what? G, A, T, T, C. So C, T, T, A, G. Okay. Now let's try to understand. First, let's see this one, or this first thing. So in this, if you see, where would be the cut present? Means we are talking about this one. Here where the cut would be present. Now my gene of interest is this C, complementary of it. Okay. So means what will happen? This is 5 dash, 3 dash, 3 dash, 5 dash. So my complementary would be like, now it's, I'm using eco R1. So eco R1 found out this, it make a cut here. It found out this, it make a cut here. It found out this, it make a cut here. It found out this, it make a cut here. Okay, like this it did it. Now when it did it, okay, this is little opposite I have written up. Let me just correct it up. Wait. Okay, so now if you see over here, where would be the cut present? Here the cut would be present at GA, and here the cut would be present over here. This thing is understood. After the cut, how this fragment will look like. So I'm gonna be writing that part. So that part will be on the next page I'm writing up. Okay. Copy. Paste. See over here how this thing will look like finally. Let me show you that. After cleaving, so one fragment and the other fragment. Can you appreciate this thing? Like, if you know that what is gonna happen here, this first G will be taken out from it. It will be out. So there will be what? It start with A, T, C, right? Whereas over here it will be starting with what? G, opposite to it. But here what would be there? These are my gene of interest. I told you now, this is what? This area is my gene of interest. This should not be cut. Okay, so I cut what? Prior to it. And it forms what? It forms something like this. This is C, 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 C. This is G, 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 G. N number of time, whatever be the gene present. So this was a gene part, my gene of interest. Coming upon the other side, if I'm going, okay. So after this, what happened? Finally over here, I have what? G. Here I have C, T. Okay, this didn't cut it over here. Okay, the cut it at G and A. T, T, A, G. So this is the fragment that has been formed. Means this part is also making, this is making bond. There is no problem. Here the proper bond is there. My genes are properly intact. This is what? This area. This is the my sticky ends. Now these sticky ends has been formed over here, and simultaneously what I have done here also, I have made this. Means the cut would be like what? This cut will form at here and at here, G and A. Let's see that after cut how it will look. Okay, so I'm gonna be making this one. Okay, I just change the look of it so that it can be fit. Okay, let's not change it or you will get confused. So it was like this, like this. Okay, now it has what? It has 1, 2, 3, 4, 5, 6. So G, A, T, T, C. Then we have C, T, T, A, G. Now what gonna happen? I'm gonna making a one cut here. I'm gonna be making one cut here. So this one cut will do what? How this thing will now look like? Let's see. We have 1 second. Okay, so now let's make the cut. So after cut, what gonna be happen? Let's see. The cut would be at this level, and that cut will open the strand a little. Means if you see it over here, this is what? This is G. Okay, A would be what? Little far off. If you will cut something, it will go away. So this A, T, T, C is present here, right? This is not a problem, right? Okay, whereas simultaneously this G is in contact with the C. Okay, so means C is also there. In front of C, there will be T also, A also, A also, and G also again. Okay, let's just make it a bigger one. Wait, you will understand better. Okay, now we have to do what? We have to cut it. Okay, I cut it up. Why we personally? So this is what? This is G. G opposite C was there, but C opposite next is T, T, A, G, right? Same way in the down, if you see, this has what? G was here. Okay, in the front, C is also there, but next to it, that is again there is what? After C, T, T, A is not there. In front of it, the things are not there. Did you able to understand what happened? So this kind of opened end thing will be formed. So if I have to just put it here. Now what will happen? See very carefully. Now here all understanding forms. If you see that this structure AATT, okay, it is what? It is opened up. AATTC. Okay, let's take it that C is also there. Okay, now AATTC can join here also, right? This part. So can I take this part and put it here? I'm showing it. That is lighter green. So lighter green part, I joined it here because it is showing that it can work like that. Whereas if you see this darker green part, in darker green part area, can you see that it is TTAAG? Can you say that its opposite will be what? TTAAG only? Which means what? Our this part, darker green part, can also come and join. So it will join at this area. So this is what? Opposite end of the green. This is the opposite end of the lighter green shade, and in between it, this area, this new thing that has been added up, this is what my gene of interest. Now you understood why do we need the same endonuclease. This is the whole concept of using the same endonuclease on both the vehicle and that one. If you didn't understand it, let me know in the comment section. I will come up with a very interactive reel for it, or the short for it. Okay, let's move on to the next pointers after this, and that is what? That is, since you you are putting this on the gel electrophores, and what is happening on the gel electrophores? Have you seen this? I have made that it is moving on it. Okay, when the current has been formed. Now the current and its direction is opposite, because electrostats said that when the current is being formed, when the electrons are moving opposite to this direction. You know this thing. So means if they are moving towards this direction, means current would be flowing in this direction. And we have what? We have the certain when the positive to negative thing, the charges are going on, we used to call it as cathode and anode. Let's try to understand that thing also. Okay, so what happens, since you cannot see pure DNA, so what, first we have to make it sure that we can see it. We put a staining on it, and then we see the UV rays lights, and there it appears bright orange in color. Okay, so this thing we are gonna write here. Since you cannot see pure DNA fragments in visible light and without staining, so staining it with ethidium bromide, which is in short form written as ITBR. Okay, followed by seeing them in the UV rays. Okay, followed by exposure to UV radiations. Then the DNA fragments appears like bright orange colored block, and it appears like ladder like thing. Okay, so ladder like, the thicker or the heavier fragment. Now how you will remember that? You know that if you are walking on a race and I give you no bag, and if I give a big bag, with big bag you can't move that faster, you can't run that faster. Same thing over here. So the ones which are heavier, have more nucleotides, bigger fragments, they will be slower at the bag, whereas lighter fragments will be moving faster. Okay, so the thicker fragments are more proximal to well, and thinner means lighter fragments are distally or distal to it. Now these different separated bands of the DNA has to be cut out from the agarose gel which we needed, which is a gene of interest. Now this band which we have cut it out from it has DNA as well as agarose gel. So we have to remove the agarose gel. So we will do what? Agarase enzyme, which will dissolve it and only the DNA will be left out with us. Okay. The separated bands of DNA are cut out from the agarose gel and extracted from the gel piece with agarase enzyme. And this process is called as eluting, taking out this part. Okay, and this process is called eluting. Remember this statement. Now the DNA fragments purified in this way are used for constructing the recombinant DNA by joining it with the ligase enzymes now. So next step would be what? We will just join them with the ligase enzyme. Okay. This we have understood. Now we will remember that there is what? A cathode and the anode. First, let me know in the comment section that how many of you know the understanding of what is cathode and anode. Now let's just try to understand. You know that since we are putting what? Electricity means putting what? Current to it. How are we going to be putting the current to it? So remember certain pointers over here. So what are the pointers? First of all, this well, where the well would be present, this would be a negative electrode. This is what? A negative electrode present here. And here is what? Opposite to it would be positive electrode. Positive electrode or negative electrode, what it means? That the flow of current would be always from the positive towards the negative side. Okay, so means what? The current flow is on this side. But if the current flow of this side, then where would be the flow of the negative ions? Towards this side. So the flow of this would be what? From negative towards the positive electrode. Opposite of that. And that's why the DNA fragments are moving. Okay, so DNA fragment movement, and current also I have told you. Okay, from negative towards the positive. On the negative electrode side only the wells are present. Moving on to the next thing. This point is over. Then the third is, I told you what? Amplification of DNA fragments of the interest, because we know what? When I'm working, someone just pushed me and all of thing was fell down, and we have to again do this whole process, which is a time consuming and expensive also. So we're gonna be doing what? We're gonna be amplifying the gene of interest, and that is with the help of a technique called polymerase chain reaction, short form as PCR. Okay, so the next third is amplification of gene of interest by using PCR, that is polymerase chain reaction. Who gave this process? This process was formed by Gary Mullis. Okay, so given by PCR technique. Okay, so it is the method for amplifying a specific region of DNA molecule without the requirement for time consuming cloning procedure. Cloning means what? We are making the similar kind of structure one after another. That also can be done. Okay, but that would be what time consuming one. This is faster. The fragment only which we needed, we will give it to it. So here the fragment would be what? The fragment would be this one. Okay, so we have to make it again and again because this has the gene of our interest. Okay, so this takes place in the appendix tube. Okay, takes place in appendix tube. Very important to remember that where this procedure takes place. Now in this technique, a small amount of DNA is put into the procedure and 3 steps takes place. We're going to be learning about it. Okay, so in this technique, small amount of DNA sample can be amplified means multiply, can be amplified many times, and sample could be anything which will have the DNA present into it. Okay, so we can utilize this for the DNA fingerprinting as well. So the sample over there could be what? The blood, WBCs in the blood, or the platelets in the blood, not the RBC, because they don't have nucleus. Okay, there could be hair, there could be any serum of the body which contains the cells into it. We can use it. Okay, so sample could be, okay, the better way, this sample could be what? It could be blood. So this is the white blood cells. Okay, then it could be what? If not the blood, then the hair. So the hair cell is like this. Okay, if not, then we have serum also, because in the body serums there are cells present. Okay, certain fragments of the cells of the body is also present, so we can get it from there. Okay, so this could be the simple, and it could be probe also for the DNA fingerprinting things. So over there we call it probe over there, that is. Okay, so now the next thing is PCR uses very important enzyme, that is Taq polymerase. This enzyme is stable at high temperature. So when we have to break the things, still it will be like high temperature also, it will maintain its integrity and it is able to work in the higher temperature, because the whole process would be at a higher temperature, because first we have to break the bonds between the 2 strands of DNA so that we can sequence it. Okay, because you know that how it opens up then sequence starts, it's like that only gonna be happening over here as well. So the very important is the enzymes which can work at it, and from where we get this Taq polymerase enzyme which do the polymerization activity. It is from a archaebacteria called as Thermus aquaticus. Okay, so archaebacteria live in the harsh environment. It is a thermoacidophils which live at the higher temperature. Someone living at higher temperature means its body enzymes would be aptly adapt for it. Clear? Uses a very important enzyme. That enzyme is Taq polymerase. Taq polymerase. Okay, which is resistant to the higher temperature. Okay, it is resistant to high temperature. Resistant to high temperature. So we can utilize it in our process now, and it is obtained from Thermus aquaticus. Okay, isolated from Thermus aquaticus. Remember it, this is what scientific name, and when this is a scientific name you have to underline it separately. Bacterium, archaebacterium. Okay, now we're going to be seeing the steps of it. Okay, so steps. steps of PCR. So number one is the denaturation. Denaturation word means what? That breaking the natural form of it. So how your DNA looks like. Okay, so your fragment was something like this. This was your fragment, right? 1 minute. So there is what? These white color are what? Hydrogen bonds, right? So the first thing would be to break this thing. So what will happen over here? Now this will leads to breakdown. So means what will happen? There will be, this is the one strand and that other will be the another strand. So these two strands would be formed. So two strands will be formed. So this is what? This is the process of denaturation. It occurs at 92 or 94 degree temperature, 94 degrees Celsius. Very important to remember, because these are what? High energy bonds. So more energy would be needed to destroy it. Okay, so these are what? Denaturation. So in denaturation, what we do? Double stranded DNA molecule becomes single stranded, become two single stranded, and each single stranded DNA functions as templates. Now means we will use it as a template, so it will make the other counterpart. So from 1 to 2 DNA thing that has been made, okay, function as template. Okay, how what we are doing it? We are just breaking the bonds. Okay, so denaturation is nothing. It is breaking of the complementary hydrogen bonds. At what temperature? 94 degree temperature. If you treat it, they will break up. Okay. Now there comes the second step, that is annealing. Annealing means what? It is priming. Priming means what? That the primer will come and attach to it. Priming. Okay. So the next step is occur at 54 degrees Celsius. Okay. Now what will happen to both of the strand? This is the first strand. This is the second. This is the second strand. Okay. These strands will have what? 5' to 3' thing, right? So this is what? 5' to 3', and this is 3' to 5'. Opposite will be there now. So what will happen? Your primer will come and join over here. Okay, so over here the primer will come on this. This is a 3' end, and here it will come on this. So this is what? Primer. This is what? Primer only. Okay, so now what will happen? Now each DNA template will form its complementary thing by putting the primer on it, because first the primer will come, then the DNA polymerase enzyme. Here the polymerase enzyme is what? Taq polymerase. Okay, so form its complementary pair with two different, see over here, this is 1, this is 2. So two different primer at 3' end of each template, because both are acting as a template. Nothing serious. The same thing you might have studied in the molecular basis of inheritance, how the replication occurs. Same thing is being done here. Okay, over there, who opened these double bonds? It is being by the helicase. Here, who is doing it? High temperature. Clear? So, template. It will form a new strand of 5' to 3', exactly same like molecular basis. That's why we are doing it right now, so that molecular basis understanding keep on going over here. Moving on to the third step. Third step is the extension or the elongation. extension, and it occurs at 72 degrees Celsius. Such a high temperature, right? So because 50-degree temperature when in the surrounding, 40 degrees is the surrounding, we feel so heat. Things about 72, which is almost double of 40, means what? Higher temperature. So we need enzymes which can work on it. So Taq polymerase will keep on adding the nucleotides now over it because it is polymerization enzyme. Okay, so now what will happen? Taq polymerase enzyme synthesize DNA strand over template, then both of them are joined together by the hydrogen complementary base pairing of the hydrogen bond and forms the two. From one DNA, two DNA has been formed. So if we had to write it in the formula that if this is the whole cycle which took place, and again it's keep on taking up. So how many cycles will leads to how many of them? We can find it by the formula 2 raised to power n, and n is what? It is the number of cycle. We're gonna be writing it up, don't worry. Okay, then on both these strands of DNA molecule, it will starts again. Okay, again PCR will start, again PCR will start, and after 30 such cycles it will form close to little less than 1 billion copies of it. So you know that it is a efficient process. So after 30 such cycles, 1 billion close, it is not exactly 1 billion, okay, little less than that, 1 billion times copies are formed of desired gene. And someone will ask what? If someone will ask like the formula for it, okay, so number of copies formed after n number of cycles can be found by 2 raised to power n. N is the number of cycle. Is the number of cycle took place. So if I say that there is one cycle only took place, so 2 raised to power 1, that is 2. That is what we said, that one DNA molecule is making only 2 DNA molecules. So if the 2 cycle has been taken place, it will be what? 2 into 2, that is 4 DNA molecules, because after this 2 will again start, 4 will form. Understood? This is easy thing. Okay, so just remember this. They can ask you this question, how many can form, in your board also, in your NEET 2026 also. Okay, with this step is over. Then we're going to be doing the one last step, that is the next step is ligation. So after that what we do, we just join it with the help of ligase enzyme. Very simple step, nothing is fancier over there. We understood it, how that has been taken place. Okay, then the rest of the steps we will be doing in the next class. Okay, so what is the fourth process? Ligation of the DNA fragment, into a vector. So what it is, this is your first fragment, this is your gene of interest, this is the sticky ends. Okay, whereas your okay, these are the two things which I will be joining it with. What I'm gonna be joining it up with the help of what? Ligase. So the ligase will do what? It will join this thing up. Okay, wait a second, I will do this. Okay, this thing, the green color thing, will join with it. Okay, is it understood? Okay, so this process is what? The ligation process. If we have to write about it with restriction endonuclease, it would be what? Similar, same. Both desired DNA fragment, you understood this concept, right? Fragment and vector DNA should be cut. Why? To form the complementary bases. So to form complementary sticky ends, which will be joined by ligase enzyme. And this is what? It is the recombinant DNA. Okay, so this is what? This is the vehicle DNA. This is the gene of interest, or it is also called as what? Passenger DNA, or it is also called as what? Alien or foreign DNA. With this, it's all about for today. For the rest of the video, you can come back tomorrow and see that it. And this is the 4 steps. Last 4 steps we're going to be doing tomorrow, which is also very conceptualizing understanding. So don't forget to watch this video, give it a like, share it with your friends. Don't forget to subscribe and hit the bell icon. And if you want a one-on-one personal mentorship with me, I am starting a batch from October itself, where your all board level questions will be done. Your board concepts and the latest papers and the trends for the boards will be there, so that we get centum in or 100 out of 100 in biology. As well as I have a special batch for the dropper and currently the school students who gonna be giving their NEET. So school students, NEET and the dropper students, they can take up that course where your boards will be also tackled. Your NEET 2026 will also be tackled. And we have an offer, exciting offers for that also. Like one of that is that if you get 360 out of 360, your whole fees will be refunded. And the fee structure is very much comfortable and easy for parents to give, because it's like the giving the fees for your local coaching institute teacher for every month. So it's not like that you gave the fees and you didn't get the efforts. You don't need to even subscribe for the next month itself. Well, isn't it crazy and not like any offers being available in the market? Well, do check it out. You can, for more details about the fee structures and the program, you can connect me on my Instagram, that is chlorophyll_classes. Well, that's all for this video. Thank you.